Carsten's third lecture, which will finish off the things he started off, and also tell us a little bit about control in actives biological systems.
So which topic one?
Topic two. I sent an email about it.
Okay, so thanks a lot, and sorry for the delay that occurred. So I would like to first, in a few slides, just summarize what I've talked about so far. There will be lots of formula on the next slides. It's not that you go through the formula that you understand them, we have seen them before. It's just that afterwards, I'm happy to share my slides so that you have everything together. This is why I put them on. There will be some remarks that I'm going to make, but it's not meant to go through in details, right? So the motivation for the things that we discussed in the last lectures was really the actin cytoskeleton. Just here a brief sketch again. So you have the cytoskeleton beneath the plasma membrane of the cell, which is often called the cortex. There is this mesh of actin filaments and many associated proteins. Then we have some special structures that are highlighted in the sketch here, like filopodia, or we have the stress fibers. Then they are in crawling cells, so this would be a cell on a substrate. It's flat. You see this big structure called the lamellipodium or the lamellipodium that really generates the forces necessary to advance the cell. And the idea was to find physical descriptions of this whole system. So first, I would like to give you some references for the biology. So I think still one of the best places to learn about the cytoskeleton is the book by Alberts. It used to be chapter 16. I'm not sure if in the newest edition it still is. If you want to know a little bit more about the fine structure, about EM data, then you can go to Sigina's paper. And there's one paper out of many others that I could have put on here that looks at the kinetics in the actin cortex. So these are just some references that you can use if you want to know more about the underlying biology and about the numbers that go together along with the actin cytoskeleton. And then, so the first thing that we talked about was these kinematic descriptions. So I'm leaving aside now these simulations that I had briefly mentioned. I guess they will be mentioned later in the talks much more extensively. And we had seen already by Rhoda these things. And so, again, I don't want to go into any details here, just to flash again how we described in a very simple approach, simple-minded approach, the actin cytoskeleton is an ensemble of rigid rods that are connected by molecular motors that can induce relative displacement between these filaments. And I only discussed the case of a bundle of filaments. All filaments are aligned. If you want to, you can't see. So there is a reference below where the framework is explained in a more general context where you can go to higher dimensions and where you also find references to other previous articles. I don't know how to take this away. So just to show that the thing here was that really the interaction between filaments of the same orientation was important. And then if you start with the periodic system where the distribution of filaments is homogeneous, and as you increase your the interaction between filaments of the same orientation, at some point you will see that you go from that the homogeneous state will become unstable and you will form these contracted bundles of filaments. And here it's just to point out that this contraction is always complete. That is, eventually you will always find a state where you have one single peak and where the rest of the system is essentially devoid of any filaments. Again, this is not meant to go into any detail, so let's just go on. Because then we went on to discuss really the theory of active gels, where we started with the conservation laws to get hydrodynamic modes. And this were in particular mass momentum and angular momentum, where I pointed out in particular that we have a contribution to the total angular momentum flux that comes really from molecular anisotropy. So this really takes into account that our filaments have an orientation associated with them. And this, like for medical crystals, gives rise to an additional part of the angular momentum flux. And this is why we take angular momentum conservation also into account here. Then the most important, the central thing was that we looked at interval production. I did not show how to calculate this, but this can be obtained from a real calculation, so that this is not arbitrary in the sense. If you want, I give some references in the slides, and you cannot see them now, but aim to see them once I've sent you. I'm sorry? That's why I have to show it.
If you can do more, I think that should give you an option.
Well, this stops only the live stream.
So then I'm wrong.
So, but
The option.
The option about.
whatever, now it's.
No, I think
Now
on
it's
top
fine.
there's, when you put the mouse.
Right, and I just wanted to point out here that in this way we obtain an expression for the entropy production that contains sums of terms, in which each term is a product of two factors, and one of the factors we're going to interpret as a generalized thermodynamic flux, and the other one as a generalized thermodynamic force. Just to say again, so the dt is defined up there, so that's just the definition of what dt is in this expression. It's not yet the full expression of what the time derivative of p alpha is. But p alpha is the alpha component of the polarization vector associated with our system. Then for the stress, the sigma, this is here the deviatory stress, and only the symmetric part of it, so everything that is not contained in the hydrodynamic hydrostatic part, which is the Ericsson stress, for which I also gave a description. I did not derive it, but just to make sure I looked again at Ericsson's paper. So this is really general also for compressible systems. So this is really the expression. And then we also had to subtract the Rayleigh stress. But in any case, this one is not so important, as we'll see in a second. And then we came finally to the constitutive equations, where we express now the deviatory stress in terms of our thermodynamic forces, which in one case is the symmetric part of the velocity gradient tensor. Then we have the coupling to the molecular field, which we can do because we have an anisotropy in our system. And then what makes this whole thing now active is the coupling to the difference in chemical potentials of ATP and its hydrolysis product. So this is how we couple the chemical energy that is released in the process of ATP hydrolysis to the mechanical part, maybe the stress in our system. Similarly, we also have an equation, and this is now the full equation for the changes in the dynamics of the polarization field. This one's given by the molecular field, and it's also coupled to the velocity gradients. Again, this is the same thing as you would have for magnetic polar liquid crystals. And then there's also an active part here, which you see again, so the coupling of P to delta mu. In principle, there would also be an equation, or there is an equation for the rate of ATP hydrolysis. And then you can write down also a conservation law for ATP, ADP, and PI. But I said that I would put myself into conditions where these three concentrations are homogeneous and fixed throughout. So there's some system from the outside that maintains these concentrations fixed. And so I don't need to consider this one. There might be interesting aspects also associated with this one, goes also along with the biochemical regulation that we are going to mention later. But for all the systems I know, this has not really been considered, and also experimentally, it's very hard to measure ATP concentrations dynamically in cells. And just these two references. So the first one is one where I think the derivation is quite nicely done. It's also very nicely done in this review article that you can find below. In both papers, there's at least one mistake, and I'm inviting you to find them at different mistakes. So reading both of them will give you the right equations. Good. Okay, so maybe just to finish this wrap up, or this reminder of the first two lectures, by some remarks. So first of all, I mentioned already that this Rayleigh stress is not really important, and that is because the cellular flows that we're interested in, they really usually occur at low Reynolds numbers. So momentum conservation is really then replaced by force balance. Then, even though the motivation for what I saw from you was the actin cytoskeleton, the same symmetries in conservation laws, but also the polar symmetry, also apply to many tissues. So you can use the same framework to tissues. And in some sense, maybe it's even better to apply this framework to describing tissues than to the cytoskeleton. The reason being that we are doing here macroscopic theory, right? We are neglecting all microscopic structures. If you think, however, about the dimensions of some cells, let's say 20 microns, and if you think about the thickness of the actin cortex, which is a few hundred nanometers, and you compare this to the dimensions of the filament, which have a thickness of 7-8 nanometers and the length of, let's say, half a micron, maybe longer, maybe shorter, then you see that you're maybe getting to the limits of where this approach should really be applicable. Yes. Yeah. No. No. So the P, and that's the thing, is really just referring to some anisotropy. It could be for a Browning cell that this is really the orientation of the actin filaments directly, but it could be also some chemical species inside that breaks the actin symmetry and still couples to the mechanic.
So how do you distinguish from the matic side?
From the matic, so if you just take the equation as I've written them down here, you don't. You distinguish them when you go to the boundaries and when you go to so what is not implicitly in here is the self-propulsion of particles. That would be another different part. So here we really deal with activity that affects the stress, the mechanical stress, but does just not lead to self-propulsion.
Okay, so it's purely stress.
It's stress, yeah. And so if you apply now this framework to tissues, so tissues are typically very much larger than the cells, and then I think there's really no problem in applying these macroscopic concepts. All this being said, I still think that if you use this framework rightly, you can get useful information. This is what I'm hoping to be able to show you later about also the actin cytoskeleton, even though the notion of macroscopic of being macroscopic might be maybe debatable. I also just want to mention two extensions that you can apply. So one is you can also incorporate viscoelastic effects. So in short time scales, the cytoskeleton, the actin cytoskeleton is elastic, and there might be more time scales than just one. But what has been done for sure is to use and to extend this framework in describing the passive properties, if you want, through a Maxwell model, so that you are elastic on short time scales and viscous on long time scales. There might be some intermediate time scales also by discussing it here. And in fact, an introduction of these viscoelastic effects is also tightly coupled to multi-component versions of this theory. So if you really, and I will add this reference later, if you're really brave, then you can go to a paper by Andrew Cullen-Jones and Pantilicha, where they try to do a full systematic derivation of a viscoelastic theory, multi-component. Sorry, Andrew Cullen-Jones. I will put the reference, I forgot to put it now. And there you just have to deal and live with, I don't know, about on the order of 30, 40 parameters. I have to confess that I've never checked in detail whether all of them are, but good. And with this, I think I will now very briefly pass on to one effect that I really would like to mention in this context, because I think it's the most spectacular effect compared to any other material that you can think of, and that is that these materials can spontaneously generate flows. And the simplest situation in which you can look at that is if you have a channel in which you put your active gel. Let's assume that the gel is incompressible, or fluid is incompressible, it's just purely viscous, and you align all your polarization vectors with the channel axis. As for the boundary conditions, we will assume that we have a strong anchoring so that the polarization is parallel to the boundary at the boundaries. And then we will also assume for these speculations, but I'm not going into any detail of the computation, that the polarization has a fixed amplitude and only the orientation changes. For the fluid itself, we would say that they slip at the boundary, so they really have free stress, or there's no shear stress at the boundaries. So this is just for which this expression has been obtained. And what does this expression tell you? It tells you that once your activity, so the difference in chemical potentials of ATP in the cytolysis products exceeds a certain threshold value, which is the one given here. At that moment, small fluctuations in the polarization are unstable. And once you have a gradient in the polarization, this will then generate flows. And these flows will positively feed back on this instability so that in the end, you get into a situation where you have a stable gradient of polarization in your system. And that is associated now with a shear flow such that in this geometry, the maximal flows would be at the boundaries, and they would be oppositely oriented, of course, in order to conserve momentum. So that's the analog of what people know in liquid crystals at the Fredericks transition. But here, mind you, there's no external force that you have to apply, but it's really through the internal processes, the chemical processes that generate active stresses, the gradient, that this comes out. And for this, you really need the anisotropy of the stress. So if you had an active stress that is isotropic, then this would not. Okay. And you see that you can have this instability not only by changing appropriately the activity, but also by changing the channel width. And for example, right, if you have some activity and you make the channel width larger and larger, then you should see these flows. Question is, does it exist? It does. And here are two examples where you can see manifestations of this instability. So this from the book of Pascal Vivaroff, so they have it took some cells, doesn't really matter what they are, that are elongated. And so you can see that you can have a structural polarity associated with these cells. They are aligned with the one is in line with the boundaries, and as you see that spontaneously they generate these shear flows. And if you go to this paper, you can see more arguments why this is really the corresponding to the instability that I've just described. A very similar phenomenon is also obtained when you confine cells to a disk. So here you again, yes, you can see that these cells, hopefully you can see that these cells are essentially elongated. And here I can show you a movie. So you see that these cells are really swirling around. And again, you can do an analysis quantifying the contributions of the active stresses versus self-propulsion of these cells to this migration. You will see that this turning around, this migration that you see is really driven mostly by active stress gradients. And you can then use such a configuration, which essentially is a +1 defect, in order to quantify all the phenomenological parameters that you have seen appearing in the equations before. Yes.
So this picture is replicated on the.
This one?
Yeah.
Also, the radius would be here 200 microns. 100 microns. The diameter is 100 microns. But you can also have this with 200 microns radius. When the disk becomes too large, then what will happen is that this defect, this +1 defect, will really break up into two +1/2 defect. Because actually these cells if you average over time, they are rather pneumatic than polar. Except for the boundary.
So if they're unipolar, does this thing flip sign? Does it flip direction?
Okay, so this one you can have them in both directions, but it's not 50-50, which suggests or just is another manifestation of the actin cytoskeleton to be chiral. And I mentioned this, I forgot to mention this. There's another extension of this theory, of course, where you look at both polar, chiral, and active charts. Polar. And to use a topological defects to quantify all the parameters goes back to Jalushinsky, who did this, or who pointed out that you can use the analysis of defects really to fix parameters for active gel, for the phenomenological description of pneumatics. Good. So this only to show you that you can have really a very interesting effect by having active polar gels, namely the spontaneous flow transition. And now I want to go back and to come again to the actin cortex, and would like to show you how you can use this active gel formalism to get some information about the cortex. And the first thing I would like to discuss is, so here in red, the cortex, so the actin is labeled, as I told you, actin is always red. And this is a paper that was written by Bas Braun, who's also now in Cambridge. And what you can see here is that the cortex is really, or seems to be a very well-defined layer beneath the cell membrane. So this would be in a cell that undergoes cell division. So you see in green microtubules that form this bipolar structure, which in the end will segregate the blue material, which is the DNA in this case, and will then in this way distribute the genetic material onto the two daughter cells. And one question that you might ask is, so what determines the size of this cortex, and how well defined is it? And to this end, schematically, what I would like to discuss is the following situation. So you have a boundary, which I will assume to be rigid, which would be the membrane. Then you have some proteins that nucleate new filaments and also polymerize new filaments. In the bulk, you will have depolarization, and then you will have, depicted in green, these molecular motors that will generate active stress. So what I like about this is to highlight that this highlights another feature that distinguishes these active gels from normal hydrodynamics. Because as you will see, this polymerization at the boundary, we are going to describe this as a flux into our system. So this is something that I think you very rarely encounter when you look at normal hydrodynamics. Usually when you have a boundary, you would say that the flux perpendicular to the boundary is zero. Here, however, since we generate new active gel, we have here effectively a boundary condition that gives us an influx of gel at the boundary. Good. And now let's look at the equation. So what I will do is, for the time being, assume that the system is isotropic. For the effect that I'm going to study, that's sufficient. And this simplifies our equations grammatically. So small change of notation. Now the actin density is denoted as C, no longer as rho. But for that, we have again mass conservation, where now we have to add something to the right-hand side, so source and sink terms, because our cytoskeleton is depolarizing at a rate kd. And of course, if we had only this one and no source term, then the asymptotic state would be very simple, but also very boring. There would be just nothing there. So there comes into account, comes into play our boundary condition that I was mentioning before. And so the current at the boundary perpendicular to the boundary will be given by a constant, essentially, for the time being, where Na would be active nucleating proteins, so proteins that help our actin filaments to grow, and Vp would be the polymerization speed. The source of the CPS is great, and we come to that. Sorry. comes now. So now force balance comes also. Now comes the constitutive equation for the stress. That's the only thing that is left to specify. So there's one part that we have seen already before. That's the viscous part. As I mentioned, I'm now in a situation where I assume the cortex to be isotropic. So there's no polarization vector, no terms associated with that. But then are the active terms. So the active terms I've written so far as zeta delta mu times something. So what turns out here is important in order to get anything reasonable, and to get anything at all, but that is related to the activity, that we have to make another extension, and that is we have to make the active stress dependent on the density. If we did not have any dependence on the density, and we just wrote here a constant, which is in principle what our theory has told me to do in the derivation that I've presented so far, then you will see that the gradients will all vanish. And so there would be nothing that comes from this active part. So there must be some coupling to a dynamic field of the stress, otherwise nothing interesting would happen. And since the only dynamic field we have in here, apart from the velocity, is really the density, we're now going to say that the stress depends on the density. Now this form, many referees don't like it. All the time that are using it, I get bitter comments and complaints about it. So the motivation for this one is that if I have molecular motors that generate active stresses, then this involves at least two filaments. So this is why I write here a term that gives me active stresses. It's supposed to come from molecular motors, for example, that involve an interaction between filament pairs. And so using mass action law, if you want, I just write here square and C. If we had only this one, then what would happen is that my system would contract, and continue to contract, and would generate a singularity at finite time. But of course, you cannot have infinite densities. So there must be something that is going against it. We call it steric interactions, call it pressure. Yeah. And this is why we add another term that counteracts this active contraction by a term that has a higher order of in C, so that for large values of C it will win. And in this way, I maintain all the quantities finite during my solutions of these equations. You could use different expressions, so you could use a function if you wanted to. So it's not really very important. Also, the exponents are not very important. I mean, it has to be at least linear here, because otherwise this contractile part again would be just a constant, and then nothing would happen. I have not tried to put just an epsilon very small, but maybe not so interesting.
So this is like a pressure check. It's an L-Ryan check. The stress is.
Oh yes, sorry. So that's missing here. That's very important. Yes, there's a delta I. The reason why I'm mixing up two things here, what I'm going to do in the first step is I'm going to look at a system that is invariant along the lateral directions, and then this is when yes. But thank you, that's very important. Okay, so now again, I will not go through any of the calculations, even though they are rather simple. We can get the analytic solution of the equations in steady state, and what you will see is that if the value of zeta is large enough, then depending on the influx that you have here, so essentially depending on the concentration that you have at zero, you will either get a profile that decays smoothly from the initial value to zero, or your profile the density, so this is now perpendicular to the membrane, so this is the z-axis. You will start from the value that you've specified, then reach a plateau value, and then abruptly go to zero. So that it goes abruptly and jumps to zero. So one jump is here for the red curve, and the other jump occurs here for the black curve. This is really due to the fact that we don't have any fluctuations in here, so we have not considered a diffusion term, for example, that you could add by hand. So I would not put too much emphasis on the fact that you have this singularity and this jump in the profile, but it shows you that as soon as you go beyond a certain critical activity, that you then get really a very well-defined cortex, which has a really a rather sharp boundary. So that's the thing that we gain from this computation, and you see already that after we had now really derived, I would say, the active polar fluid equations, we now get away and modify them as we see fit in order to describe real systems. Okay, but now if you look at a real cortex, you will see in some instances at least that it's not really as static as the previous picture might have suggested. But instead what you see is that there are large fluctuations that you see very badly here, but if you look at that data, you will see that there are protrusions coming all the time out and retract again. If you measure the thickness of a cortex in a cell that does not show these protrusions, then still there are fluctuations of the thickness of the cortex thickness. And this has been measured in the book of Giardina and Reggio, so with Mathieu Piel, where they use magnetic beads. One of them they insert into the cell, or the cell really just through phagocytosis eats it up. And then you have a second bead outside. And these magnetic beads, you put them into a magnetic curve, and thereby you can really measure very nicely the distances. You can also exert forces on the cortex and pinch it. And then you see here, over the course of 300 seconds, that you have large fluctuations in the thickness of the cortex. So I think these are really the best, most precise measurements of the cortex thickness. I think are really better than optical measurement. And you see that the cortex thickness varies between 200 and 700 nanometers. So there are huge fluctuations. And these fluctuations depend on activity. They depend on motors, but even more importantly, they depend on the polarization and depolarization dynamics of the filament. So this just to mention that the cortex is really not as static as the previous picture might have said. Yeah.
So now you point out that it's a very dynamic system without something over. I think it's quite fast. In my mind, there's a bit of a gap between that and this picture of fluid that flows. How is momentum transfer if the thing is constantly being turned over?
So what you will see in a second is that also the system that I was looking at before generates flows in presence of the turnover. So you will have turnover, which essentially makes the system go back to some reference density with this plateau that we've seen. But in addition, with the stresses and isotropy, you will see that there are also lateral flows. I don't think that this is incompatible with momentum transfer, because the momentum flux has just to occur on time scales that are, I guess, faster or shorter than for turnover. I don't have a timestamp on this one, so I don't know how fast it is. But we see that these occur on quite long time scales, right? Time scales of 10, 20 seconds. For the time being. Sorry? No. We will introduce it now. Because these are still the same equation as before, but we should really now for the part here. So what we will have here, so this is now a numerical solution of this equation, where at the membrane we introduce a friction between the material and the in the boundary, even though that is not strictly necessary in order to see the phenomenon that I'm going to show you. Yellow here means that we have reached the plateau value. So this would be a stationary state as we have computed it before. Blue would be a part where there's no actin. So again, the membrane, if you want, would be on top, and then you look down into the cell. And what we have done here, so the first thing we had just put it by hand, but this is now a computation where we have really introduced a polarization field, and I've just not described it here. And so this means now we have anisotropic stress. And what you will see is that as long as the stress along the lateral direction is equal or inferior to the stress along the vertical direction, this state is really stable. But in the opposite case, then what you can see is the following. So you see that the cortex breaks up, and then now you see the emergence of these protrusions that go into the cell, mind you. Because we have here, and we have here really a rigid boundary, so there's nothing that could protrude outside. And so you see that you can generate, in this case, really fluctuations in the cortex without any biochemical control. So this is really just the intrinsic dynamics of the system. And if you look at Lyapunov exponents, there's some indications that this is really chaotic dynamics, and this will go on for as long as you are willing to run the code. So again, this might be, but this we don't know, might be underlying the fluctuations that you see on the left. And it might be, and again, for the time being, we don't know, that they are responsible also for the formation of these structures that have been obtained in the book of vitriol. Where here now you see actin in blue, no longer red. So this would be in a cell that is crawling. You see that there are protrusions going outwards. Again, this is something that we cannot describe in here. You see also that they persist towards the inside. Whether left and right are the same phenomena remains to be seen. But it shows that at least on some very superficial level, you can generate the structures that we see in the theory. You can also observe them in experiments.
You said this stuff seems to show live exposition. It seems to be in them. That it's chaotic.
Yes.
Because when I'm looking at them, they look like there's a sort of typical size of these objects. So this is just an artifact.
So there is a certain distance because you will form new protrusions only once the two protrusions are far enough apart. And it forms also rather readily after they have distance. So there is some characteristic scale in there. That's true. So if you count the numbers, you will see it's always between 3 and 5.
Okay,
But it's still chaotic.
okay, but there's no membrane already.
It's just a boundary. It's just a fixed boundary. So the computations with the membrane are coming. Wait. Okay. Good. So, but this is not the only interesting structure that the cortex can show. So what I've shown you so far was to describe the cortex perpendicular to the membrane. Now I would like to describe the cortex lateral to the membrane. And this here is the giant egg. So I think it's a frog egg. Maybe see if you can remember. And what you will see here is that the actin is again labeled, but only the filamentous actin, not the monomers. Again, it's not red, but this brownish color. But you will also see that there's another component labeled, and that is Rho. So Rho is really a class of proteins, so-called small GTPases, that affect the activity of the number of proteins that regulate the actin cytoskeleton, particularly its assembly, but also contractility of the system. And I think I just let the movie speak for itself. This is not the only movie that you can find. There are others that also show these or similar kinds of patterns. We often see target waves, spiral waves, and in these large systems, they can be observed quite frequently. This is just one quite nice example of it. And so one question you might ask yourself, so what is going on here? I would also like to mention that this is only one instance of waves. There are other instances of waves, maybe not as, or certainly not as regular, not as beautiful, but still also as important. So here it's a human immune cell, has been put on a substrate. Again, well, actually here it's not actin that is labeled, but a protein associated with actin doesn't really matter too much. But here you see also that you see waves emanating from the center in a kind of target wave pattern, so they're not very stable because the cell is moving. But you see here that these waves, as they reach the boundary of the cell, they push the boundaries forward. And so the idea of Orion Weiner and his colleagues was that these spontaneous waves, that they might orchestrate the active cytoskeleton network or the active myosin network in order to get the right organization to obtain motion. So here you see that this cell is not moving really directionally. It's very erratic. But that's for an immune cell, maybe not a bad idea. If it doesn't know where to go to, it has to just patrol our body in order to see whether there are pathogens. And you probably don't want to just walk in one direction. Okay, so the question is, how can we understand these patterns? And what has come out over the years is that there is a feedback between raw activity and actin. So as I said, raw, which itself can be in active or an inactive state, and there are very different kinds of raws, so I just call it primarily raw. This one can activate and inactivate proteins that control actin. And that's represented by this arrow here. But then what has been found out over the years is also that once you have F-actin, then it will inactivate raw. And this is illustrated by this blunt arrow there on top. And then in addition, there's evidence that raw can activate itself. And this really are basic ingredients that you need for an excitable system. So there's no mechanics involved, it's just chemistry. And this you expect to generate waves. And indeed it does. Yeah, it's, I mean, I would rather link it to Fitsunagumo to make it even more simple, but I guess it's a very complicated version of that. Also here, I mean, there are many things and reactions in the immediate steps push another rug. Right? I mean, it's not raw would directly affect actin, raw would affect maybe performance would then nucleate and polymerize actin, then actin would recruit some factor that would then act back on the activity of raw. So it's much more complicated, but I don't want to get into these details here. Which raises, of course, one question, and that is, so I'm going to show you, I guess, a wave soon. Are these waves the same waves that we see in the experiments? I mean, I guess what we do know is that these waves are coming from an excitable system, and that's what I'm presenting you here. So we have here the inactive and active forms of these nucleators, how I will call them from now on. So you have the self-activating term, you have the inactivation of the nucleators through the cytoskeleton, and we have here just a pure diffusive reaction diffusion equation also for the nucleator for the actin. And this can generate waves, and you can put them into cells and they move, all fine. And now I'm running out of time. So
yeah, so
we started late. Do you allow me 10 minutes?
Yeah.
Maybe you want to do this thing
Yeah.
If you have a question about the previous slide, ask me later. Because what I would like to point out now is more that all the states that I've shown you before, they really affect the whole cortex. But if you look at this picture here, where again actin has been labeled, there are isolated structures. So these dots, and these dots are not just noise, they are really structures that have an internal structure. Namely, you have actin, which is indicated through this other protein, cortectin, in the center, and then it's surrounded by a corona of row activity, active row. So this is now row C, it doesn't matter the detail. But what you see is that these are really structures that are not just random there, but they are really, they even have a, people call a function, because they are linked to what people call invadopodia. In fact, the cell on the left-hand side is a breast cancer cell, and this one tries to invade surrounding tissue, which it would do by standing protrusions that are called invadopodia. And the first signs of these invadopodia are these structures that you see here. And so you see that there are really patterns or substructures of the cortex that are localized, and this we haven't seen so far. So is this then just biochemistry, or is there something else? And I would again argue that you can get this from cytoskeletal self-organization, where we now put back into our system the mechanics. And in addition, we couple our species that control our cytoskeleton to the flows that are generated by gradients in the active stress by replacing the diffusion current for the nucleators by this diffusive advection current that you see. And then this time I've done it correctly. So that's the topic part. And there is some experimental evidence that really these proteins are convected by the actomyosin network. So this is not just our fantasy, but this term really, this coupling really exists. And then what you can see, if you now solve the equations numerically, you can see these states that are localized. So this, at first sight, might be very similar to the states that I showed you in the very beginning, where we had in this kinematic description just this contraction. But there are two differences. One difference is that the zero concentration, by the way, you see in the dotted line is the these raw proteins, whereas in the black line this is actin. So as in the pictures I've shown you before, you have actin in the center, and you have this corona of the raw protein. But that's not the point I wanted to make now. The point I wanted to make now is that really you have this localization on top of a homogeneous background. So you do not destroy the complete cortex, but you just add onto the cortex a structure. And the second thing is that when I go to larger and larger systems, I should change this at some point. So and really look at this different way, then you see that the and that's not so nice to represent here, but what you can maybe see is that the homogeneous part really remains unaffected. And also the state that is generated in this localized area really remains the same, which you cannot see really, but if you believe me, then you can maybe see that this extension of the state gets smaller, and this is just because I have extended the scale. So these states, they are really independent, or rather independent of the size of our system. And this is if you go to very large length, then this is really the state that you would see. Again, you see actin inside, the row outside, and then what is depicted here is also the flow that keeps these structures together. So through the active stresses, we really have the flow coming from the outside and assembling our material. Good. Yeah, this is just to make clear again the analogy between the two. And I will not show you the underlying mathematics, which is quite nice, though, if you like nonlinear dynamics, the physics or the mathematics of localized states. So I will skip that. I cannot jump forward. It's all very nice, but okay. So, but this I want to show you. And that is to show that they are not only stationary localized states, but they can also be oscillatory. So maybe many of you know these oscillons. When you shake granular matter in the right way, then you see also these nice localized structures that jump up and down. And you can also have chaotic states that are also localized. It's just to show you a little bit the variety of states that you can obtain. What I do want to discuss in 2 minutes at least is what is really needed for this localization, because these are now the equations in one dimension of what I've shown you before. And the question is, which of these terms are really necessary? Which of the elements, because there are now quite a lot of them in there, are really important? So again, we have mass conservation of the active cytoskeleton with the two assembly and disassembly terms. We have the equations for active and inactive nucleators, and they can be activated and inactivated, but there's no production of them. And the currents are always the convective currents due to the flow that is generated by the active stress gradients. And there's also diffusion. And I should mention that the active nucleators are in fact those that are bound to a membrane, so they diffuse not as fast as the ones that are inactive, which are not bound to the membrane. And then we have here our force balance equation, which has the dissipated part here and the active part on the right hand side. So the first thing to check is, do we need active contraction in order to get these localized states? And in this description, yes, you need them. So without this term, you use them. Even though there are RH and diffusion systems that can produce localized states, in this case, it's not possible. Do you need the biochemistry to? So do you need to couple the actin to active and inactive nucleators? Yes, you need. Otherwise, it doesn't work. Do you need the conservation law? Which conservation law really mean the conservation of the total number of nucleators? And this one you also need. And I can discuss this later if you want. This will take 4 or 5 minutes to make it mindful. Then you can ask, but do you need really a separate actomyosin field? Do I need to have the C field separately, or can I just, sorry for this word, but enslave this field to the dynamics of the nucleator? So if alpha and A minus ADC is very much larger than the rest, then I could just express NA in terms of C in terms of NA. Is this maybe also sufficient? And that indeed is sufficient. So you can simplify your system in this way and still get the localization. Do you need these nonlinear terms in the biochemistry? No, you don't need that. So the simplest system that we found based on the initial one is this one that can still show the localization. However, the localized states then look differently because they do not show these internal structures anymore, but are rather flat states. And this, to just end with a few pictures, I want to show that what I've shown you for these localized states is all in one dimension, but you can have them also in 2D. So this is the most boring extension to 2D. It's just radially symmetric. This could be stable, but in this situation, it's not stable. You see that this looks almost like cell division, right? Embryo with a single cell, but it's not. It's really the equations that we've shown before, and it's not meant to be describing cell division or embryonic development. It's really just to show you that you can also have, in this case, localized states with a more with some internal structure, and then you can have even more interesting states that are dynamic, where you have this is now the final state, where you have some more or less stationary part in the center, and then you have these blobs going around. But that's more for fun than for anything else. And I'm going to explain why it's called the Plumsock state. But I want to conclude a little bit. So, namely, that mechanical coupling can generate localized states. I think that's quite clear. I have not talked to you about the static snaking. That's details for people who are interested. I can talk about it later. Localized states, well, they could be an alternative to liquid-liquid phase separation for localizing molecules. And I think to work out what is the relation between these two concepts is an interesting thing to do in the future. And now that we are going to write and speculate that these localized states, they could be used by a cell or by an organism to position disclination points. In disclination points have been shown, at least in Hydra, to be important for generating hydrogen morphogenesis. And this is just to make a little bit advertisement for Dan who did these computations. But with this, I would like to thank you for all your attention. I would like to acknowledge everything that I presented on the localized states was really done by Luca and then Oriane. She worked out a few of the bifurcation diagrams. And this is all part of a collaboration with Daniel Brunner in Zurich, Olivier Platz in Bern, and Daniel Levin in Strasbourg, funded by the Fonds National. And with this, I'm really finished now. Thank you.